talk about molecular diagnostics testings and imprinting disorders and related growth disturbances, I will begin with a general overview on genetic testing and human genetic testing to bring you all together to summarize the current knowledge on diagnostic testing, and then I will switch to the specific and complex testing of imprinting disorders. In case you have any questions, please use the chat, as Sandra has already explained. But now let us immediately start. So when we talk about genetic testing, we do not only have to consider the methods and their limitations, but we also have to ask, why do we need genetic testing? Why is it necessary? What is the benefit for the patient and their family? Until recently, human genetic testing was just a further tool to confirm a clinical diagnosis and to estimate recurrence risk. But as recent years have shown, genetic testing results are becoming more and more relevant for personalized treatment, and the identification of the molecular defect of a clinical phenotype as early as possible enables us to offer these treatments and avoid severe courses. But not only the early diagnosis in pediatric endocrinology is a major milestone in genetic testing, but also the identification of the molecular cause in adults with endocrine disturbances and adults with diagnostic or disease. Furthermore, personalized treatment has already been implemented in oncology, and the modern genetic tests are the basis of these developments and options. So within the next minutes, I will try to give an overview on these different strategies and developments. So I will give you a general introduction in genetic testing, then I will switch to congenital imprinting disorders, and I will focus on the molecular defects. Finally, I will show you some strategies of genetic testing. Sorry. Just a short reminder. When we talk about genetic testing, we are talking about the mutations we are addressing. We have different levels of mutations, and we can discriminate between chromosomal aberrations like in trisomy 21 or DiGeorge syndrome with small deletions. Then we have the classical monogenetic diseases characterized by point mutations. And then we have the imprinting disorders with a very specific and special type of mutations or molecular defects. These are the epimutations and imprinting disorders. Different tools, a broad range of tools is available for human genetic testing. We can either target, as I already said, chromosomal aberrations with by chromosomal conventional analysis. We can use array-based CNV detection tools, the microarrays. Then we have, of course, PCR as a standard and basic tool of genetic testing. It is complemented by Sanger sequencing, targeting single sequence variants or single monogenetic diseases like cystic fibrosis. But then, of course, we have established and implemented next generation sequencing. And we have additional methods which help us to further define and characterize molecular alterations like MLPA or quantitative PCR. However, all these techniques have advantages, but also limitations. And this is a very rough overview on the limitations we have with the different methods. Chromosomal analysis address copy number variations and numerical aberrations down to 5 megabases, but the resolution is very low. Therefore, we have several years ago, fluorescence in situ hybridization has been established, but the situation had improved when microarrays were developed and implemented in routine diagnostics 20 years ago. On the other hand, we had techniques in the past addressing single nucleotide variants, CNV detection tools with very specific tests like for thrombophilia or hemochromatosis. Then we have Sanger sequencing, which is up to now the gold standard of DNA sequence analysis. However, both techniques, CNV detection by simple tests and Sanger sequencing, only cover up to 100 or 1,000 base pairs. But what is missing is, or what we have to cover is the lag between these very high resolution techniques and the low resolution techniques, and this is achieved by the implementation of next generation sequencing. But all these techniques address conventional or classical monogenetic and genetic alterations. So we are talking on molecular defects on DNA level. The situation is different in genomic imprinting disorders. Genomic imprinting itself is an epigenetic phenomenon that causes different expression of genes in a very parent of origin specific manner. This means that we have up to 100 genes in the human genome, which are specifically expressed in only in a monoallelic manner. That means we have genes which are expressed only from the maternal allele or from the paternal allele here. And as you can see here, in this case, in this example, the paternal allele, the paternal copy of the gene is silenced by methylation groups, whereas the maternal allele is active. Vice versa, we have the situation that the paternal allele is silenced and is active, and the maternal allele is silenced by methylation groups. This is only a very simple example of how genomic imprinting is working. However, we have always to bear this hypothesis in mind. We have to remember in all the following slides that we have genes which are monoallelically expressed, and the expression depends on the parent of origin contributing the allele. Genomic imprinting is also organized in a life cycle of imprinting, meaning that we have an erasure of the imprinting marks with the primordial germ cells. All imprinting marks are erased. Then the imprinting is reestablished during oogenesis or spermatogenesis, and this methylation signal, these imprinting marks are then maintained during the imprinting cycle of life. And as you can imagine, this this fine balanced pattern of imprinting marks is very prone to disturbances. And therefore, in fact, we can observe several imprinting disorders, several human diseases, which are characterized by the disturbance of this fine-tuned genomic imprinting balance. Therefore, I want now to switch to congenital imprinting disorders. We have already heard some of the following aspects from Irene Netschin in a great talk several weeks ago in the webinar on Silver-Russell syndrome, but I think I will repeat some of our major messages just to remind you why all these considerations are important for genetic testing of imprinting disorders. Imprinting disorders are characterized by specific clinical features, and on the other hand, by molecular genetic features. I will talk about this later. Let us start with the clinical features. Imprinting disorders are generally clinically heterogeneous, but they share several findings. Several of the imprinting disorders are characterized by asymmetry. Several show growth disturbances. In the majority of imprinting disorders, these are growth restriction features, but we also can observe overgrowth, like in Beckwith-Wiedemann syndrome and Kagami-Ogata syndrome. Then we might have metabolic disturbances. In Angelman syndrome and some others, we have a developmental delay or mental retardation. We have specific dysmorphisms, abdominal wall defects, and in Beckwith-Wiedemann and Kagami-Ogata syndrome, again, we have increased embryonal tumor risks. However, these are only examples of clinical findings we might have, but each imprinting disorder is characterized by a specific combination of all these features. And therefore, we can observe a clinical overlap between the disorders and the patients with these disorders. And sometimes it's very difficult to diagnose patients with imprinting disorders due to the clinical overlap and the unspecific features they share. Additionally, several molecular genetic features or molecular genetic alterations in imprinting disorder patients are characterized by molecular mosaicism. This means that not all cells are affected by the molecular defect, but that only some cells show or carry the epimutation or the imprinting disorder molecular alterations. And the degree of the mosaicism might influence the clinical severity of these disorders. Interestingly, as you can see here in this slide, we also have very interesting findings in twins. The majority of twins reported with imprinting disorders were monozygotic, but they were clinically discordant. That means these brothers are monozygotic twins, but I think it's obvious that these patients are clinically discordant. Currently, we know 13 imprinting disorders. Here you see an overview on 12 of them. The 13th has recently been described. It's a precocious puberty syndrome associated with the chromosome 14 locus, but the other 12 are shown here. Some have already been identified and described in the past. I think Angelman syndrome and Prader-Willi syndrome are well-known imprinting disorders, also called microdeletion syndromes, because many of these patients show a microdeletion in 15q. But there are also some other well-known imprinting disorders like Beckwith-Wiedemann syndrome and Silver-Russell syndrome. Then we have the new imprinting disorders like Temple syndrome, Kagame-Gato syndrome. I mentioned that syndrome before. And we have some very rare or currently probably undiagnosed imprinting disorders, which are very new, but these here are characterized by monogenetic changes, DNA sequence alterations. These syndromes here, precocious puberty, Schaaf-Young syndrome, baromental retardation, they do not show the whole spectrum of molecular alterations, but they only show DNA sequence alterations. Here's some clinical feature pictures of patients with imprinting disorders. Examples are Silver-Russell syndrome, Beckwith-Wiedemann syndrome, Angelman, etc. But I think you probably more than about these clinical pictures than I do. I mentioned that the clinical overlap between the different disorders is a major challenge in the decision process which locus should be tested. Here, for example, we have an adult female, an adult patient with Silver-Russell syndrome. She was diagnosed at the age of 40 years because she reported that as a child she was diagnosed as Silver-Russell syndrome. Then we looked for her pictures from childhood, and then we decided, well, this is a typical child with Silver-Russell syndrome. As an adult, it's not that obvious, this phenotype, but on the basis of the pictures from her childhood, we decided we should perform Silver-Russell syndrome. In fact, we could confirm that clinical diagnosis by molecular testing. But this is a typical Silver-Russell patient, but then we have here a patient with a Beckwith-Wiedemann also exhibiting the typical signs of Beckwith-Wiedemann, but here another patient which was diagnosed as Silver-Russell syndrome. It turns out that she had a Temple syndrome, molecular Temple syndrome, a chromosome 14 alteration. Another example referred for genetic testing for Silver-Russell syndrome. It turned out that he was molecularly a Beckwith-Wiedemann syndrome, and so on. So all these examples show you how difficult it is to diagnose an imprinting patient based on clinical signs. We have, of course, a major group of imprinting disorder patients with a typical clinical signs of, let's say, Silver-Russell syndrome, for example. But on the other hand, we have a huge number of patients with a very unspecific and even overlapping clinical phenotype, and therefore it is often difficult in clinical practice to decide which test should be performed or should be advised. Now I switch to the molecular alterations which can be observed in imprinting disorders. The typical imprinting disorders like Prader-Willi, Angelman, Beckwith-Wiedemann, they show 4 different types of molecular alterations. The most prominent molecular finding is probably the maternal or the uniparental disomy. Here is shown a maternal UPD as an example. Maternal UPD means that both chromosomes of a pair have been inherited from the same parent. And the problem in this situation is that in case you have a monoallelically parent of origin specifically expressed genes, in that situation you have disturbance of the fine-tuned balance of expression. meaning that, for example, a gene which is solely expressed from the paternal allele becomes silent. Vice versa, we have an overexpression of a maternally expressed genes because now both chromosomes derived from the mother. The other three, then you might also have deletions or duplications, and in any case, you have, as I said, you have a disturbance of the balanced expression of a gene. You have the same in point mutation. In point mutation, the problem is you might have a very specific gene affected by the molecular alteration and a monogenetic point mutation, the missense mutation, frameshift mutation. But the problem in this situation is that an imprinted gene is affected, and in case here, as shown here, in case the paternal gene is affected, which you might need for expression, then it's not expressed due to the unbalanced, due to the disturbance by the mutation. Then the typical finding in imprinting disorders is the fourth group of molecular alterations. These are epimutations. Epimutation means that you have an aberrant methylation pattern at a specific differential methylated region in an imprinted region. Here you see, for example, a hypermethylation. In that situation, the paternal gene, which should be active on the paternal chromosome, is silenced due to a methylation group. This is called hypermethylation, but we also observe hypomethylation with a hyperactivation and overexpression on the maternally expressed genes. So you can have different situations disturbing these imprinting domains, and you can have different types of mutations. In case you offer genetic testing for imprinted disorders, you have to be aware of these four different types, and you have to ask, does your method you are applying, does it cover all these four groups of molecular alterations? So let us, why is it important to subdiagnose, or not only to diagnose an imprinting disorders, but to categorize the molecular defects? The reason is, from a human geneticist point of view, that we want to define recurrence risk. Here you see uniparental disomy as one of the four molecular alterations which can be observed in imprinting disorders. And as you can see here, the four major imprinting disorders show different ratios of uniparental disomy among the molecular spectrum. In transneonatal diabetes mellitus, it's a very frequent finding. In Prader-Willi, it at least accounts for 20% of patients, and so on. And the problem is, in case of uniparental disomy, that you have some imprinting disorders which in which uniparental disomy might be associated with an increased recurrence risk due to chromosomal aberrations or familial chromosomal aberrations. I think it's meanwhile well established that in case of Prader-Willi and Angelman syndrome, you have to be aware of Robertsonian translocations on chromosomal aberrations, which might be predisposing for uniparental disomy. In other imprinting disorders, it's very low, but in case you have evidence for the chromosomal alteration from the family's pedigree, for example, then you have to be very careful and you have to ask for conventional or fish karyotyping. Second group, deletions, duplications. The problem with deletions and duplications in imprinting disorders might be that not only the imprinted region is affected by the deletion or duplication, but that the deletion might also comprise larger regions, including other genes, and thereby the phenotype might become altered. That means you might have a different phenotype than the typical imprinting disorder phenotype because more genes are affected than only the imprinted gene in chromosome 7, 11, etc. So you have a correlation between the size of the deletion duplication and the phenotype. And the phenotype might be mimicked by a very large chromosomal duplication deletion of an imprinted, close to an imprinting disorders. And of course, again, you have to consider the alteration, the nature of the alteration for genetic counseling. And as I said, the phenotype might be modified due to further genes which might be affected by the copy number variation. Then we have monogenetic mutations as the third group. These are rather rare, or currently rare. We know UBE3A mutations in Angelman syndrome, where it contributes significantly to the molecular spectrum in Angelman syndrome patients. But in the other imprinting disorders, it's rather rare. We already know CDKN1C loss of function mutation Beckwith-Wiedemann syndrome, accounting to up to 5% of patients, of sporadic patients. In families with Beckwith-Wiedemann, it's even larger, it's up to 50%. In Silver-Russell syndrome, these variants are very rare, but I think the future will show whether they will account for a larger ratio of causes. Last but not least, the fourth class of molecular changes in imprinting disorders. These are the epimutations. That means these are isolated methylation disturbances and without any obvious reason for a genomic or obvious genomic change causing the epimutation. This is only partially true because meanwhile there is an increase meanwhile of reports on alterations, molecular alterations indirectly causing epimutations. That means we meanwhile know some patients with Beckwith-Wiedemann syndrome and loss of methylation of the imprinting center region 2. And in some of these patients, the KCNQ1 gene is affected by mutations, and the lack of transcript of KCNQ1, this is long QT gene localized or encoded in 11p. Mutations in the KCNQ1 gene cause an aberrant transcript of these genes, and this indirectly causes a loss of methylation of ICR2. So, to summarize, epimutations the molecular reason for epimutations might be unknown, but there is an increase of reports which help us to understand how epimutations are caused. And therefore, we have to consider that in genetic testing and in genetic counseling. So an example is shown here for imprinting disorders. These are Silver-Russell syndrome, Beckwith-Wiedemann syndrome, the two syndromes associated with 11p alterations. And I've chosen to show you Beckwith-Wiedemann and SRS, Silver-Russell syndrome, because they are good examples to illustrate the complexity of the molecular findings, and this has consequences for genetic testing. First of all, as I already said, we have a very broad spectrum, a very heterogeneous spectrum of molecular alterations and the same imprinted locus in 11p. We have the 4 different classes. With a locus heterogeneity in Silver-Russells, we also have chromosome 7 and chromosome 14 affected. We have a broad clinical heterogeneity. It's not that easy to diagnose these patients clinically. We have overlapping phenotypes. We have mosaicism, at least for UPD11 and the epimutations, which might make the diagnosis more difficult because they modify the phenotype. We have unexpected findings. We have multi-locus imprinting defects. And finally, the question we have to answer as human geneticists, we might have increased recurrence risks. Mosaicism, I think we should repeat it again and again, because mosaicism is really a major challenge in imprinting disorders, because they have an impact on the clinical severity. And as you can see here in this example, we have the patients. In this patient, we have analyzed blood cells, we have analyzed buccal swab DNA left and on the right side, and you see differences here. It's more prominent. We have a very broad degree of very great differences in the distribution of the mosaicism between blood, buccal swab on the left side and on the right side. So mosaicism is indeed a major challenge. Then we have multilocus defects. Currently, we do not know the contribution of MLID to the phenotype, but we think that MLID probably have a more important influence than we currently know. Multilocus imprinting defect means that we have not only one imprinted locus affected, but that several others show aberrant methylation. But interestingly, as we know from the current imprinting disorders, we probably have, or we might have, a clinical dominant locus. For example, here in Beckwith-Wiedemann syndrome, all Beckwith-Wiedemann syndromes identified with MLID have been identified on the basis of imprinting center 2, loss of methylation, and then it turned out by further tests that they were affected by MLID. However, this might be this distribution here, or these observations might be influenced by an ascertainment bias because we started with a Beckwith-Wiedemann diagnosis or a Silver-Russell diagnosis. Then we identified the disease-causing epimutation, and then it turned out that it's MLID, but I think we have to look broader to identify patients with unusual phenotypes with another pattern of multilocus imprinting defects. Why do I mention MLID, multilocus imprinting disturbances? Because MLID gives us some ideas how imprinting is regulated and how epimutations are caused. Meanwhile, we know that assisted reproduction techniques might have an influence on the methylation pattern of an embryo, and several MLID patients have been born after assisted reproduction. Then we meanwhile know that we have to consider cis-acting genetic factors, but they only will affect the specific loci. I have already mentioned the case here in Q1, long QT variants affecting the Beckwith-Wiedemann syndrome, locus IC2. But then meanwhile, we have also identified so-called trans-acting genetic factors. These are factors which play a role in the imprinting cycle of life. They are responsible for the setting, the maintenance of imprinting marks. Examples are shown here. There are the number of examples is meanwhile growing in the literature, but some classical findings are shown here. We meanwhile know that ZFP47 mutations cause TNA. It's also a factor which is involved in the maintenance of methylation, and mutation in that genes, they cause transient neonatal diabetes mellitus and MLID. Deborah McKay and Karen Temple and their group have already shown that several years ago. Then other groups have shown that the factors of the subcortical maternal complex in the early oocytes might be responsible for an aberrant methylation. These mutations are called maternal effect mutations. It was a group from Esther Meyer and Eamon Mayer who reported for the first time that these genes, maternal effect mutations in these genes responsible for the maintenance of methylation in the early oocyte, that mutation in these genes might cause an aberrant methylation pattern in the offspring. That means not the child is the patient, but of course clinically the child is a patient, but molecularly the mother of the patient is responsible for the alteration, for the molecular disturbances in her child, because the lack of NALP2 and other genes, NALP2 protein in the early oocyte cause aberrant methylation. And interestingly, these genes or mutation in these genes are not only associated with imprinting disorders, but also with miscarriages, aneuploidies, and preeclampsia. Therefore, in case you diagnose a child with imprinting disorders, it would be reasonable to ask for the family history, and in case you hear something about miscarriages, aneuploidies, etc., then you might consider to ask for a NALP, NALP4, a NALP2, NALP5, etc. testing. Now, finally, I want to go to the genetic testing itself. Before we start genetic testing of imprinting disorders, we have to ask several questions. First of all, is the phenotype specific? Should we start with a specific test for Silver-Russell, Beckwith-Wiedemann, etc., or should we rather apply a broad imprinting disorder test addressing several imprinted imprinted loci. Then you have to bear in mind which type of mutations are known. Does my test cover all these epimutations? Do I have to apply additional tests? Is a locus-specific test applicable? Or should we rather apply a multi-locus test? I mentioned that before. Then the different methods have different limitations and advantages. You should be aware of that. Of course, it's the job of the laboratory offering you as clinicians, the genetic test. Labs should inform you about the limitation and the advantages of the methods. Then we have to ask, do we have mosaicism? Has mosaicism been observed? Should we test further tissues than blood? Are additional tissues necessary? Then later for the reporting, has inheritance been reported? Are additional analysis required? And here you see once again the frequencies of the different molecular defects and the 8 prominent imprinters. I again want to show you that slide because I want to remind you that we have different frequencies for the different molecular defects and the different imprinting disorders. And for each disorder, each syndrome, you have to consider the frequencies, and on that basis you have to start or have to define which test should be performed and which strategy should be followed. And corresponding to the tests addressing genomic changes, you also have a similar methods for methylation specific tests. We have methylation next generation sequencing, which we'll cover in the future. I think in 5 years, we will probably apply methylation based next generation sequencing, and perhaps you might address all questions you want to solve, and you can also cover all 4 different groups of mutations and epimutations in imprinting disorders. But currently we have single locus tests, and we have methylation microarrays, but they are, as far as I know, not implemented in routine diagnostics. Here you see the different the advantages and limitations of the different tests which are currently used in imprinting disorders testing. And I want to focus your interest on MLPA because, as far as I know, this is currently the widely used test in diagnostic, and with that test you can identify or at least 3 different types of 3 of the different molecular alterations which you might observe, and the major advantage is that you can discriminate between the 3 different types of molecular changes. And finally, you can multiplex MLPA to address multi-locus testing. Here are my examples for MLPA. In case you are more interested in that technique, you can visit the MRC Holland site, which provides us with the MLPA test. Here are examples. In the upper lane, you see a Beckwith-Wiedemann patient with a normal copy number run but a hypermethylation. Here you see that the deletion in 11p, shown in the upper panel, causes a methylation shown here in the lower panel. So multi-locus testing, I have already mentioned that. Multi-locus testing, is that really useful, or is it necessary? Is it sufficient to test to apply specific imprinting disorders tests? In case of Prader-Willi and Angelman, I think it's not necessary to apply multi-locus testing because Prader-Willi and Angelman syndrome are closely linked to the chromosome 15 locus, but in the other disorders, you might consider multi-locus testing because Beckwith-Wiedemann, Russell, and the others, they show a very broad range and a broad overlap, clinical overlap. And therefore, multi-locus testing might be helpful as a first-line test to diagnose these rare, different and rare alterations. So therefore, multi-locus testing might improve and fasten the testing strategy. Here's some examples. As you can see here, this patient has an MLID, but both have this is an isolated IC1 loss of methylation. But as you can see, both patients show the typical Russell phenotype. Here, a patient which has been initially diagnosed as Russell syndrome, but As you can see, he does not show the typical phenotype, and it turned out he had a uniparental chromosome 7, a maternal UPD7, a typical Silver-Russell finding, but additionally had the Temple syndrome, gain of methylation of the chromosome 14 locus, so a mixed phenotype. Multi-locus testing, again, why is it needed? It's needed from the, it should be discussed from the human geneticist point of view, because these families with multi-locus imprinting defects, they have an increased risk for miscarriages, for recurrence risk for children with imprinting disorders, and perhaps you might also continue with the reproductive counseling. In these families, it has been shown that oocyte donation might be a helpful treatment for the reproductive questions. Yes, this, I think I have already shown you this patient. I will skip it. Another point for Becker-Wiedemann syndrome, some of you might know that 10% of UPD11 patients, UPD11 accounts for 20% of Becker-Wiedemann syndrome patients, and among these 20% of patients, 10% again have a mosaic paternal uniparental diploidy, meaning a genome-wide uniparental disomy, and these patients have a very increased risk to develop tumor even in adulthood, and they also show very severe causes of tumor development, and they often die in early childhood. Therefore, it's very important to identify these patients. In case of UPD11, you always have to perform further tests to exclude uniparental disomy or genome-wide uniparental disomy. Mosaicism is a challenge. I've already told you that. So, and therefore you might remember human geneticists reports that they write, while mosaicism cannot be excluded. This is a very important message for you. You can never exclude mosaicism for epimutations or UPD11 in Becker-Wiedemann syndrome. And in case you are sure that your patient has an imprinting disorder, then ask for testing of a second tissue, because it might be that you will miss the typical molecular finding in blood cells, but that you might find it in hair roots, in buccal swab DNA, in fibro and skin biopsies, etc. So therefore, always ask for testing of a second tissue in case of a negative first-line test. Final outcome of all our genetic tests are the reports, and I hope that you will all get concise and precise reports from your human geneticist. Otherwise, ask them to become concise. These reports should correspond to the European standard norm ISO 15189. They should include standardized and internationally recommended nomenclatures. They should have a clear interpretation. This test confirms the clinical diagnosis or does not confirm the clinical diagnosis. The report should include an advice for genetic counseling. Whether it should include an advice for clinical management is in discussion. I think not all countries agree with that advices for clinical management should be included in genetic reports, but it depends on the national policies. Finally, very important, you have to mention the limitation of the method. But I want to remind you again, genetic testing can only confirm a clinical diagnosis or carriership. It can not exclude it. The diagnostic algorithm for imprinting disorders should always start with the methylation analysis and dosage analysis. So even in Prader-Willi syndrome, you should not start with a FISH or conventional cytogenetic analysis, but you should start with a methylation test, because the methylation test will give you all information about other alterations and the imprinted loci. Here you probably know that several consensus guidelines have been published on Silver-Russell, pseudohyper, and Becker-Wiedemann syndrome. In case you're interested, I can provide you with the literature later. You can send me an email for that. Yeah, but in case you have negative methylation testing results, the question is what to do next. You can apply disease-specific tests. For example, in Russell syndrome, it's recommended to test chromosome 20, to test chromosome 16, perhaps. But I think finally you will come out, or you will finally have to decide whether you want to perform next generation sequencing, either panel-based sequencing or whole exome sequencing. And I think several published studies also from other monogenetic disorders have shown that NGS will significantly increase the diagnostic yield in these disorders. Therefore, NGS probably is the next step after negative testing results from the first-line approaches. An example is shown here. It is relevant to diagnose or to continue with the molecular diagnosis to identify the molecular cause of patients. Here is an example of a patient which has initially been presented to our institute with a clinical diagnosis of Silver-Russell syndrome, and finally it turned out that he was homozygous for a mutation in the Bloom gene, and this is very important because Bloom gene is a tumor predisposition syndrome, and of course you need this information for your clinical treatment. This is an overview on genetic testing of congenital disorders in general, and I think you might know this from our joint paper from the Endo-ERN, just to remind you that genetic testing is not that easy as it might be regarded, and that you might have different results. Of course, you hope to get information on the pathogenic variant, but of course you might get information on variants of unknown significance. You might have incidental findings in case you apply next generation sequencing. Therefore, be careful in case you apply next generation sequencing. Yes, but at the end, a short summary. First of all, the take-home messages should be that that methylation-specific testing should be the first-line test for. In case of imprinting disorder testing, you have to be aware, or at least the lab should be aware of the chances and the limitations of the test they apply. You have to consider mosaicism. It might be that you will ask for testing of a further tissue in case you are sure that the patient has a clinical diagnosis of an imprinting disorders. Molecular subgrouping is relevant for clinical, genetic, and reproductive management. We have at the beginning, but we know that epimutations can be caused by genomic causes, and these genomic causes are associated with an increased recurrence risk. In the future, we probably will apply more than we do it now. We will apply whole exome sequencing and perhaps whole genome sequencing and long-read third-generation sequencing. Nevertheless, all these tests have to underlie a quality management system. We have to establish, or we have to carry out quality assessment schemes. And why do we do these tests? Why do we need the very precise molecular testing? We need it for our future genome medicine, meaning that we have a personalized medicine based on very precise molecular diagnosis, which I have already mentioned in the beginning. Finally, I would like to thank, of course, all of you for your patience. I want to thank the audience for your attention. Then I would like to thank the families and the patients, because without these patients and their families, I think all our work is not possible. Thanks again. I want to thank my colleagues here from Germany, and the international colleagues, particularly from our former COST network. And they all contribute to the knowledge we currently have and which I could present today. Thank you for your attention. And now I'm waiting for questions via the chat. Thank you. Is it important, in your opinion, to send DNA of patients suspects of imprinting disorders to an expert lab in this field? Yes, Irene, I think it's an important question, an important point. I think the first-line diagnosis might be performed locally in the different countries and the different labs. Here in Germany, for example, we have private labs and university labs, which perform the first-line testing, but in case you identify patients with epimutations or other rare molecular disturbances, then it might be very useful to send these samples or these patients to expert labs. You can identify them in our network, for example, or on the national level. Otherwise, you can contact Irene Nechin, Karen Temple, and other members of our former imprinting network. You can ask us to give you information on expert labs in your country. But I think it's important because we further have to characterize these patients. As you might have seen from the multi-locus patients, it's necessary to identify the genomic cause, because for the family it's important, because these disturbances might be associated with increased recurrence risk for miscarriages, etc., tumor risks. Therefore, it's important to further characterize these patients. On the other hand, it's not only the diagnostic point of view, but of course, we would be happy to bring all these samples together and these families because these diseases are very rare and we need more information on a broader basis of information and data. Yes, so we would be happy in case you forward us samples for further analysis. I think all of our team, of our members of our former COST network. Thank you.